Yeah, there are, I guess, two messages. One of them is that indeed were able, after optimization of this CRISPR-Cas technology, we were able to sterilize cell cultures in the laboratory from infectious HIV-1. And by doing so, we actually discovered novel mechanism, how the virus is inactivated. So that's nice for a scientist. But of course, the main question is, can we develop this in a therapy? And so I showed great success in the laboratory. But talking about treating a patient, the first question is, where do we want to deliver this therapy? Where is this virus hiding? And actually, despite many years of research, we don't really know where this so-called HIV-1 reservoir is. And there was a very prominent paper 2 years ago, published in Nature by a group from France, Montpellier. And they published a new potential marker of this reservoir. The interesting thing is that many top laboratories in the last year, they have jumped on this and they could not reproduce the finding. So by now, most people in the field don't believe in this marker anymore. But in the meantime, we also started working on this new molecule. And actually, we have become a firm believer of this molecule. And again, that's nice for science, but it's also very nice because you want to use this molecule to target the reservoir where the virus is hiding. So on the one hand, we have big success with CRISPR-Cas in the laboratory, and we slowly are moving now towards the in vivo applications, which is still very difficult. Yeah, so of course, there's always the question of safety. So despite the success that we had in cell culture, this was with a system where the CRISPR-Cas system is delivered for good and it's active for forever. That's probably why it is so effective. But of course, concerning safety, I would not easily propose to do the same thing in patients, because if CRISPR-Cas is hanging around in the body of a patient, and it will cleave some DNA, so now and then you can expect there will be potential off-target effects, toxic effects in the long run. So we actually have now to move to totally different CRISPR-Cas technologies that only will deliver CRISPR-Cas for a transient action. It has to inactivate HIV-1 and then it should be inactive for the rest of the patient's life. So that's one thing we are working on. And of course, targeting of this reservoir, in this case the CD32 cells. We think these are the cells to target, but how to get there, that's another important thing that we plan to work on. And we will employ one of the well-known vector systems that people use in gene therapy, lentiviral vectors. But we have to change them such that they will specifically go for these CD32 positive cells. So two of the things that have to be done, but there's probably much more.